Okay, we're going to look at now the fourth and final type of point mutation that can change the protein sequence and cause problems with the proteins. Here again is our original sequence with our MET, ARG, SEER, SEER. The mutation this time is going to be an insertion mutation. And what that means is a nucleotide is going to get added where it's not supposed to be. So this little arrow shows where this nucleotide is added. And then, of course, the RNA is going to just copy the DNA, so it has an extra nucleotide in here. So to see the effect of this on the protein, we're going to have to first draw our codons. Okay, so here in our original sequence, we found our AUG, and that coded for MET, then our CGA, which coded for ARG, our UCU, and our AGC, which code for serine, and then we had our stop codon. In this new RNA that had this inserted nucleotide, we're going to find our AUG just as normal. But now, remember our CGA was our next codon. But sticking this A in here means that the codon has completely changed now to CAG. And this extra A, which used to be part of that codon, has been pushed to the next codon. And here we have our future codons. So you can see that this completely changed the sequence of everything after that insertion. So when we go to, again, we have to look at the effect of this on a protein by translating it. So we know we have our MET, and we get to our CAG. This used to be CGA, but now it's CAG. So we look at our codon table, where CGA codes for arginine, CAG codes for glutamic acid. And now our next codon, which used to be UCU, that A was pushed into it. The UCU is still here, but that A pushed it over. So now we have AUC, and AUC codes for isoleucine. So notice how every single amino acid after that insertion is going to be out of frame, and therefore is going to be incorrect. Okay, so that type of mutation is called a frame shift mutation because we've shifted what we call the reading frame. We've made all of the codons out of sync. Frame shift mutations are very, very bad on a protein because now all of the amino acids after that insertion, it also works with a deletion. If you shift the reading frame of the mutation, every single amino acid downstream of that mistake is going to be incorrect. So probably that protein will not be functional.